Our next speaker is Sheri McLane, owner and president of Interag Services, IAS, Laboratories in Phoenix, Arizona. She is a certified agronomist, certified crop advisor, certified professional agronomist, and a valuable resource to the agricultural community of Arizona. Skilled in providing fertility and water recommendation for optimum economic production of both field and vegetable crops.
My name is Sheri McLane. I am the owner, the certified crop advisor, the certified agronomist over here at IAS Laboratories here in Phoenix. And we're just going to discuss today about soil testing. You know, what do the numbers mean? Why should I care? Because a lot of people get results from laboratories and they really don't know how to read the report, and sometimes they're too embarrassed to ask. And so hopefully this will help clarify some of that information for you. Alrighty. So why should we really soil test? Well, it's part of our best management practices strategies, right? So a lot of us are switching over from the conventional practices over to conservational practices. And so part of these conservational practices is we are trying to get a whole area map of our crop nutrient program management, right? So we know what nutrients a crop requires. We know what nutrients are in either our manures, our compost, our fertilizers we're putting in the ground. We also need to know what's in our soil. We need a baseline. And that allows us to not over-apply, to reduce the amount of nutrients we're putting in the soil. And by putting down just what the crop needs, hopefully that can save us money because we're not wasting money in unnecessary fertilizer. It can also save us time. So if you don't have to put that extra application of fertilizer, that's less labor, less gas, less wear and tear on your vehicles. So all that kind of works together to kind of help micromanage and get the farm working more of a fine-tuned machine. And we do this by using science. We're not going to just go out there and guess. We're not going to look out there and say, well, I usually put down some zinc this time of year, whether my crop needs it or not. We're going to use science. That's going to allow us to be proactive and not reactive. It's going to let us know when the plants start to show signs of deficiencies internally using science before we actually see it. And so that way you can put down what you need, when you need it, and keep everything under control. By doing science and using testing, you're able to discover your acute problems as well as your chronic. If you were to do some soil testing and plant tissue testing over several growing seasons, you're going to start seeing typical issues you have on your farm. Maybe you need to add gypsum because your calcium value is always too low. Maybe you've got a zinc problem, a boron problem. You'll see these acute problems and you'll be able to incorporate that into your fertilizer management plan from the get-go and get it budgeted out so you don't have any surprises going down the future while you grow your crops. And this also allows you, to see any acute problems. You might have a magnesium deficiency, or maybe you've got a toxicity that we need to work with, and you'll be able to see those problems by analyzing the soil and the plants before the yield gets reduced, before your crop starts to show signs of problems. And so that's a great way of using soil testing and plant testing. This allows you to fine-tune your operations, get it more efficient, and hopefully in the long term, you can either increase your yields or at least quality. How many times have you gone to the grocery store and you've seen, spots on the bottom of the lettuce? That's a boron deficiency. You get a melon, 3 days later, you shove your thumb through it. That's nitrogen toxicity, also a boron possible deficiency. So by just adding what your crop needs, you can increase your yield, increase the shelf life of the products, and increase the quality. That's what a salesperson is going to say to help you really do testing. But the truth of the matter, why do you test? Because you don't want to fail. No one likes failure. And if you don't test, you have a possibility of your fields looking like this photo here. If you look on the left side, the before, that is, if you were to say, what's wrong with this field? You might say, oh, I got salt burn. It's just too many heat units. It's the end of my crop. You see it's nitrogen deficiency. Reality, this is a molybdenum deficiency. The before is 7 and the after. That is just a difference of 3 days after application. That is a difference between losing your crop 2 weeks before harvest and having success. And so by doing testing, you can reduce and prevent your failure. You need to do this because you might put out maybe a $500,000, $10 million loan of credit to grow for your season. You want to be able to pay that back. You want to make sure using testing that you can secure your investment so that you can keep continuing farming for generations to come. This also preserves your livelihood, right? So if you fail, you can't put your money back. You need to make sure that you're providing for your family, maybe you have children, maybe taking care of your parents, family members. No one really ever farms alone. You have workers. Those workers are counting on you. Those families are counting on you. So you need to do testing to help preserve your livelihood, to keep things going, and just to ensure that, hey, I'm going to be okay. I'm going to get some harvesting and hopefully find these problems so you don't have failures. And really, that's why the people I work, that's why they test. So they have success or better success. Now you're thinking, okay, maybe I should do some testing. Well, how do I sample? You know, yeah, people have services for sampling. We sample for people. There's fertilizer representatives. They go out there, they'll sample for you. But if you want to do it yourself, it's not difficult. First thing you need to do is figure out what your goal is. What question are you trying to ask, right? Are you doing this before you plant as a pre-plant? Do you have a field that just doesn't perform as well as you know it can and you want to correct that? Maybe you've got a bald spot in the middle of the field. Maybe something happened, the manure skipper double applied somewhere, equipment broke down, something got dumped there. You've got a bald spot, you want to get that up to par so the whole field is really producing really well. You got to figure out what your goal is and then come up with a plan. And here's kind of an example of a plan. I went on Google Earth. Anyone can do it. It's very simple. You can get a picture of your farm if you don't already have one. I'm sure most of you do. And you can see where your fields are and then just kind of grid it out. So for example, in the blue rectangle for number 1, if you were doing a pre-plant sample, you might take the entire block because you're going to fertilize it the same. You're going to irrigate the same. You're going to plant it all the same. Let's just get a baseline. See what needs to be applied before you see to put your transplants in. And so you'll just use that as your one whole sample. Most people might say keep it below 60 acres, but my theory is if you've got an area that's 120 acres and you're not going to change your fertilizer program for that whole space, you might as well treat it as one big unit. Why waste your money and waste your time? Now, there's other situations of how you might have your goals for split up samples. Let's look at the orange area, the spot for 2 and 3. So per se, you just took area 2 out of beets and area 3 just came out of spinach. Different crops leave behind different crop residues. Perhaps you're doing no-till situation while the residues on top. Per se, you're going to be split planting those areas. You might want to figure out what's going on differently. And so in that case, you might split the samples 2 and 3, even though it's the same field, if you're willing to treat it and fertilize it differently. If not, you might as well put it all together as one sample. Again, why waste your time and your money? But at least try to get a baseline as to what's going on. And then the third example we have is on the bottom. That would be area 5 and 4. Say that's one big field, but area 5 just isn't doing well. You struggle every year to get a stand, the plants are stunted, something's just not right. And you can tell by this aerial photo, it's not disease, right? You can actually tell by the washout marks, the plants are growing based off of the mineralogy of the soil, of the landmarks that were there, whether it was 5 years ago, 10 years ago, before the fields were put in. And so if you're willing to make the nutrient adjustments for area 5, we can sample those differently, figure out what the problems are, and then get those corrected so then the whole field can be treated the same in the future, and you'll have more optimal growth on both sides of the field. So just depending on what kind of goal you want to achieve, what kind of answer you're trying to find, you're going to break up your field sampling in that manner. So you might think, okay, I know how I'm going to put it together. How do I sample? We usually recommend walking the field diagonally and taking many subsamples along the way, mix that together to make one sample. Don't just take it from one spot. Like I said, what if you had a gopher hole and something happened in that area and you have bad numbers? What if the manure skipper overapplied there and you have bad numbers? You want to walk the field diagonally and subsample from about 20 to 30 different places, mix that together to make one sample to get an idea of what the whole field is. And yeah, it's hot out there, you're very busy, you may not want to do this yourself. That's why there's companies out there, the officers, or maybe your fertilizer rep or your CPA, they might be willing to do this for you. If not, you can always hire a high school student, teach them how, and they can also do it for you as well. Other people might suggest, hey, walk the field in an X. You kind of get a little bit more coverage of the field, a little bit more area. We've done that here in the lab, comparing numbers, and the numbers come out very similar. Other people just say, walk the field randomly, hodgepodge. The main goal is just don't take it from the corners in one spot in the middle. Take the time, gather soil from a little bit from everywhere, and now you have a better idea of what your crops are going to be going through. Now, some of you might be having nut orchards, maybe you've got fruit orchards. And so obviously you're not going to take the soil from, 20 feet in from the furrow, and things of that nature. You're going to be walking those fields necessarily diagonally, and you've got vegetables growing out there. If you're growing trees, like nut orchards or something, when you do walk your fields, grab soil from the drip line. You can kind of see the drip line here on the sides, on the edge of the tree. That's where most of your watering should be happening. That's where a lot of the nutrient roots are. That's where a lot of the fertilizer is applied. That's where all the action is happening. So we want to take the soil sample from that area so we can help better find out what the trees will be taking up during the growing season. When you do take a sample, try to use something like a probe or a shovel. Don't use a pipe. Pipes have zinc in it. We test for zinc, and that will contaminate your sample. Auger probes work great. Like I said, shovel or spade works good. Just make sure it's not rusty. You don't want that metal getting your sample. And like I said before, when you walk the fields diagonally, you want to take 20 to 30 subsamples. We usually grab soil from the 2 to 8 inches of depth, just because, that top inch, you're going to build up of salts from evapotranspiration. Your roots aren't typically at that top inch. And that way you have a better idea of what's happening in the actual root zone. If a lot of you are switching to drip, a lot of times you want to try to get close to the drip tape because that's where the fertilizer and the nutrients are going to be. And then that way you have a better idea of what these plants have access to. Once you grab your 20 to 30 samples, mix it together, throw it all in one bag, preferably a plastic Ziploc bag. In fact, these are perfect for sampling. Paper blind bags are plastic, Tupperware, Ziploc bags from the grocery store. These work fantastic. They hold the moisture in so the soil doesn't fall out. If you grab a paper bag and the seam is not plastic lined, the moisture in the soil will actually loosen the seams, and when you pick your bag up, the soil goes everywhere. So we don't want to do that. Plastic containers work great. If you don't have anything, call the lab that you're comfortable with using. They should be able to ship you some containers. I do want to say, don't use these. I know it seems a little silly to put this slide in, but believe it or not, we've gotten samples with this packaging. I've had plant samples in Cheez-It bags show up at the lab with Cheez-It still in the bag. Just last week, a gentleman brought in a soil sample in a salted peanut jar. He's convinced that he saw the salt out. He wanted us to test it anyway. But still, we test for salts. We're testing for calcium. We're testing for boron, which is in glass, all these different things. Please don't use contaminated containers that will not give you the accurate results you need so you can make the appropriate decisions that you need to make on your farm. When you do submit a sample to a laboratory, any laboratory, most labs should have a chain of custody up on their website. If you can, please fill one out. At least give them, your name and phone number so they can contact you. If you put a sample at field ID, so you know it's going to be called when the results come back to you, you write down the test you want, the crop you're growing, it really helps that laboratory do the job the first time correctly for you. many times, even here, I'm sure other labs have the same problem, samples will actually show up with no information. We have to go off the address on the box, and sometimes it's just a UPS store. And they're not going to remember who you are when you drop the sample off. If you're in a hurry, if anything, just toss a business card in there. It has your name and number in there, and at least that way, we can call you, find out, okay, what are you trying to achieve? What are your goals? What do you want tested? And if you're not sure what you need, talk with the laboratory they're using, work it out. They'll help figure out what you need to achieve the goals you're trying to get. So now you have it testing, you know how to map, you know how to sample, you've got your chain of custody filled out. You're probably thinking, okay, what test do I request? There's always so many options on these websites. I don't have time to call, just tell me what I need to do. So in my opinion, I would recommend doing a complete soil test before you plant or right after you plant. We call it pre-plant soil test. Whatever lab you should get, make sure it has all these elements. The reason why I'm saying make sure is I've seen reports from many different laboratories when I've done consulting for other growers, and believe it or not, some of these reports don't have the micronutrients on them, which you need for flowering. Some reports don't even have a nitrogen value on there, which is bizarre because without nitrogen, a lot of the times all the elements fall apart. So make sure that you have a pH, a salinity, know what your salt problems are, the major nutrients, right? The nitrogen, phosphorus, potassium, those secondary nutrients, calcium, magnesium, sulfur, all those micronutrients, right? Iron, manganese, zinc, boron, molybdenum, boron, sodium. Get an idea of everything that you have because your crops are going to require all these things, and we want to get rid of any limiting factors. If you're trying to build up your organic matter, switch to more organics, things of that nature, you can always add on an organic matter test. If you're having drainage problems, get a texture test. Find out, is it a texture problem? Is it a salt blockage problem? Those things also help. If you're trying to switch over organics, want to build up the soil biology, ask for like a microbial profile analysis that gives you like 6 functional groups. It tells you all the microbiology. Those are the ones I recommend. If you are on a budget, at least do a complete soil test to get a baseline of what this field has, so then you know how much fertilizer to apply so you don't waste your money in excess fertilizer. During the growing season, if you choose to do further soil testing, not tissue testing, usually I recommend just doing a nitrate soil if you've got a crop like an onion or corn or something of that nature that doesn't have a petio, the stem that we can help. People usually test for nitrate purposes. If you're having a salt problem, you want to manage it during the growing season, just have the salts tested. Look at the sodium, look at the overall salinity. You don't have to do a complete every single time. If you want to see how the organic matter is building up over time because you're building different crops, your crop rotating, you got pasture, you want to see how the root mass is doing, you can always do an organic matter value. If you want to see how the microbials is building up over time, now you switched over to maybe organic, so you're trying to do something of that nature, you can always have the microbial profile test done. If you choose to do a microbial profile test, I just want to forewarn everyone, it is a snapshot of what's happening the moment you pull it. And so to really compare apples to apples, if you're going to test it more than once, try to sample the soil same number of days after you irrigate it, because these microbials are going to be more active depending on the temperature outside, the moisture content. Did you just fertilize? Did you add organic material? All those different factors are going to play on these microbials. These are live critters, constantly moving. You know, it's like chemical warfare down there. They're eating each other. It's just madness. So try to at least sample for microbial profile under similar conditions, or at least the same, you know, how many days after you've irrigated, just to keep things more consistent when you try to compare something. It's not fair to try to compare if you hadn't irrigated in 3 weeks and then you irrigated 3 days ago. The numbers are going to be different because of dormancy in the microbial biology. So I know I said a lot of things here. So ideally, if it was my farm, I would do a complete soil test before I plant with a possible organic matter and microbial profile if I want to monitor the biology. And then during the growing season, I would actually test the plant itself to see how it's doing, to find out what is it taking up, what is it having a hard time with. And when you have a pre-plant soil, the lab that you use should be able to look at your pre-plant results, whether it's a month prior, and be like, okay, you are okay in these elements, you struggled in zinc, assuming you applied it, you shouldn't need to add, phosphorus, add this element, you add fulvic acid instead. They should be able to look at all the numbers, look at all the ratios and trends, and figure it out for you. If they can't, find a lab who can, or an agronomist or a CCA or someone with that knowledge who can help you. And so I was also asked to discuss, there's different types of nutrient tests out there. A lot of people talk about water solubility analyses, what's plant available in the soil, and it kind of confuses people because they're not sure what exactly is the difference. What is this information actually telling me? And so I just kind of want to go over kind of quickly. If you have a plant available or plant soil test, complete soil test, that's what's going to tell you what's available for the whole growing season. I want to say the entire growing season. That tells you the nutrients that are going to be going to the root that dissolve in the irrigation water by mass flow. And that's also going to be the nutrients that are available as the roots grow and intercept it. So you're kind of getting both the mass flow nutrients, which are the water soluble ones, and the ones that are going to be available by root interception. The levels that laboratories have are based off of published research, and there's actually proficiency programs that agriculture labs participate in using the same methods to make sure that everyone is on the same page. And so that's why I like the available for the growing season, because I'm getting everything a whole picture. Now, if you're having a salt problem, you need to know more about salt management, then I would recommend maybe a water soluble analysis. That's only going to tell you what's available at that one irrigation. The water soluble test, basically you're just taking water, making a paste, and seeing what dissolves in your water. So anything that's soluble is going to show up in analyses. The micronutrients don't show up very well in it. The phosphorus doesn't show up very well in it. It's your salts, those positive cations, the calcium, sodium, potassium, magnesium, nitrate. That's what will show up on that analysis. And so if you're, like I said, if you're trying to do more salt management, you can always do that analysis. When I see numbers on the complete soil test, I can see salt problems for the whole growing season, not just that one irrigation. But if you want to micromanage each irrigation event, do the water soluble. If you choose water soluble, and I can't stress this enough, no one does it, but I wish they would listen, submit a water sample with your soil. Ideally, your irrigation water should be used with the same soil from your farm for water soluble. That'll give you a true picture of what's happening for salt management. If you don't, the SOPs laboratories have say to use DI, which is filtered water. That only tells you what's happening in the soil, does not take into account what the chemical reaction is happening between your irrigation water, which could be salty as can be, and what's happening with the soil. The pH of your irrigation water will also affect the availability of what comes out of that irrigation, out of that soil. So if you do water soluble, please, for the best answers, submit your water with it so they can mix the two together to give you the most honest answer. So I kind of mentioned it a little bit already, but just to kind of reiterate, for salt management, really, you have three options. Option one, the one my brain is programmed to do, and it's not like, before I step back, that one of these options are better than the other. It's whatever you're comfortable with in interpreting the values, or your consultant is best understanding that it should be used. I like option one. It shows me both the short-term salt problems you're going to have, as well as the long-term problems that you have. So we can kind of nip both in the bud. A lot of salt management products that are out there, they like to do the water soluble because they're trying to fix each irrigation event. And so the water soluble is more for that, for your short-term problems. Option two just gives you numbers, and you kind of have to interpret it and do it math. Other people like to do base saturation. I haven't talked about this one, but base saturation is very similar to water soluble, except we do the math for you, or the lab should do the math for you. The water soluble is all in parts per million. Base saturation is in percentages, so you can just see the percent in relation to all the other salts. And then you can just grab the numbers and go. You don't have to do the math. And so I like base saturation a little bit better than water soluble, but really it has to do with your comfort zone. And so those are your three options. Choose what's best for your comfort zone. Another analysis people always ask is texture and cation exchange capacity, or CEC. I get this a lot for people who are trying to find out how much nutrient, how much organic matter to put in the ground, things of that nature. And so if you do a texture test, the texture and CEC are very closely correlated. A lot of lab reports will give you a calculated CEC value. I don't like the calculated CEC value. In Arizona, our calcium value is around 3,000 to 7,000 parts per million. And I find when the calcium value is very high, it actually inflates the CEC value 2 to 3 times the value. So instead of reporting a value of 7, I might be reporting a value around 21. And that's not good data for you. You can't make honest decisions on that. So if you really want to know what you should have, if you do a texture test, is your sand, silt, clay, it's very correlated to CEC. And as you should know, if you have a lot of sand, your soil cannot hold a lot of nutrients, it doesn't hold a lot of water, so you usually have to fertilize more often with less, or you water less but more often. Opposite is with clay. If you have a lot of clay, that's what the nutrients hold onto, that's what the water holds onto, those more micropores for it to cling onto. The charge is different, and so you actually add more fertilizer but less often with clay. It's very much correlated with the CEC. It's all based off of nutrients holding things. So if you have a high clay content, you're gonna have a higher CEC, low clay content, low CEC. And so if you're not sure which to do, in my opinion, if you do a texture value, it kind of will help you figure out what your CEC values. Arizona is sandy loam. Technically, our CEC values are going to be between 7 and 12. And here's a quick chart. You can kind of see it on the bottom. You can see the different soil textures and the CEC values, and they're all very well correlated. For those of you who might have tested that in the past, you might notice some correlation. So in my opinion, don't waste your money if you don't have to. Test for one or the other, but usually the two are correlated with each other. And then you got your microbial profile. I'm just throwing this out here in case you're wondering, okay, if I do have a microbial test, what should I expect? You should be able to get the count for your nitrogen fixers, your yeast and molds, your aerobic bacteria, your anaerobic bacteria, your pseudomonads, and your pseudomyces. There's so much different biology in there. We don't necessarily tell you all the different individual species, but we'll kind of just clump it together in different groups. So you have an idea of what you have. And then we have interpretation charts. So you can say, okay, are these numbers good and bad? And if you're trying to build up your soil biology, this is a good balance, or a good way to start. So what does this all mean? Let's put this all together. And I'm gonna kind of have to go really quickly through this. I apologize, I talk a little bit long. A soil report will show all your problems, your deficiencies, your toxicities, if things are out of balance, things of that nature. And so I'm just gonna kind of go through this very quickly. So if you don't keep up, I apologize. We can always go over this slower at another time. Wherever you get a soil report done, there should be a key somewhere. So, and here in red, you can see it. You've got levels of very high, high, medium, low, very low. If you see something that's very high and or very low, don't panic. This doesn't mean you have a toxicity problem. This usually means if it's high or very high, with the exception of sodium, this usually means on an individual element basis, let me stress, individual element basis, you don't need more, there's more than plenty. If it's medium, yeah, your crop will probably do a little bit better if you add more, but you gotta look at the economic value. It may not be, if you put the extra, say, zinc in, are you gonna get return on investment? If you put the extra nitrogen in, are you gonna get the yields you need to cover that cost? So you gotta look at that from the medium perspective. If it's low or very low, with the exception of sodium or your salinity, that typically means this is your limiting factor. If you don't add this, your yields can be reduced. And this is kind of why I'm going to talk about pH real quick. A lot of people are always fixated on pH. This is where a lot of people try to aim their pH around a 6.8. That's because that's when all the nutrients are the most available. The wider the bars of each element, that's how available things are. This is the pH of Arizona soil. You can see we usually have boron deficiencies, iron deficiencies, copper deficiencies, zinc deficiencies. Phosphorus is pretty tiny. And so people are always fixated on pH. I don't fixate on pH. I'm bringing this up because a lot of people are trying to get their pH super low. And if you drop your pH too low, you can actually cause nutrient toxicities here in Arizona. We actually have manganese blooms here. And so I try to tell people, yeah, biology likes a little bit more acidic, but be careful. You might release the kraken. You need to be careful. If you do have a lot of nutrient deficiencies like phosphorus and you have a super high pH, you can reduce the pH to make those nutrients more available. My goal is usually a pH of 7.8 because that's when the nitrogen is the most available and is not so tied up. It's also more cost effective than dropping it super, super low. So just keep in mind, high pH, nutrients will be tied up more, low pH, more nutrients become more available, but be careful, you don't want to cause a nutrient toxicity. And then you have different elements. You've got your salt. When people say the word salt, don't freak out. It's not necessarily a bad thing. Salts are positive cations. That is your calcium, magnesium, sodium, and potassium. And the things that you want to look at when you look at a report for your salts is one, you want your sodium below 300 and you want your potassium below 900. If they're not, you will have plant burn. You'll get an outer ridge burn or yellowing on your plant. And you want to make sure your calcium magnesium ratios are balanced. So a lot of times you might have enough magnesium in the soil on an individual element basis, but you have to add more to balance the different elements. And so that's why I don't like having an absolute black or white value. This is how much I need. It's all about balancing those ratios. Plants uptake nutrients based on concentration and concentration gradients. So you can be very high in these elements, but if they're out of balance, you'll show deficiencies. If you're low in calcium, a lot of different things happen. One, you can get crusting. If it's just a 1 or 2 millimeters thick, it'll happen. When you don't have enough calcium, you'll have ponding water, you'll have deflocking of the soil particles because the sodium and the particles just kind of splurge everywhere and then glue back together. And then as your plants emerge, they can snap. The hypocotyl roots can snap because it's too hard for them to break through the soil crust. So you want to make sure your calcium is not low. If your calcium is also very low and your salinity is low and your free lime is low, that's a sign that you're going to have low oxygen in the soil. That's going to cause you to have stunted plants and you're going to be more susceptible to disease and to root rot. So you want to make sure those numbers are in the medium or high range, with the exception of you don't want too much salinity, you'll burn stuff. And then you got your calcium magnesium. I kind of mentioned it quickly already. You want to make sure this is around a 10:1 to 21. If it's too wide, you don't have the soil moisture, your plants will be stunted. If it's too narrow, you don't have the oxygen in the soil, your plants will be stunted. So you want to have it balanced. Sometimes if it's too wide, if you've got adequate boron aids the uptake of calcium, magnesium, that can help get those elements into the plant. So you kind of want to look at those three all together in one situation. Sodium, you don't want it too high. Too much sodium, even if you water your plants can't get the water, they get dehydrated. The saltier the water is, the more energy that plant has to have to pull that water molecule up into the roots into the plant. And the more salt it is, the more pressure, and if the plants just don't have that energy to do so, they'll wilt and will start to die. Also, if the plants uptake sodium, the sodium is swapped out for calcium, magnesium, and then you have cell leakage, and then your plants are going to start turning yellow. Potassium. You want to keep that potassium around 140-ish to 900. Too much potassium, it'll actually block the uptake of calcium, magnesium, and it's going to burn your plants. If it's too low, your plants are going to fall over. They're going to lodge. So you got to find that nice sweet spot. I'm going to go through this a little bit quickly. I apologize. I'm running over time. I'll try to wrap it up here in just a moment or two. Your estimated computed percent sodium percentage, that tells us if your salts are balanced. If they're not, the number is going to be very, very high, and then you can have ponding water, and you're going to have bad infiltration on your, and you're going to cause root rot. Your overall salinity. For most vegetable plants, you want this to be below 3. For your grains, tomatoes, forages, pastures, 4 to 5, depending on the plant. If not, you got to irrigate and flush that salt out. These are the 4 elements that influence that salinity the most. And so you can have high nitrate and high sulfur, and that's going to cause your plants to burn. So just keep in mind, too much of one thing is not good. So we want to monitor them as a whole package. You can actually be adequate in all 4, but as a unit, they can actually cause your plants to burn because it's just too much salt, and it will actually cause them to yellow. If you overwater, sodium, salinity, and nitrate will actually drop substantially. So your soil reports will show us that. Your plants in your soil don't lie. If you have too much watering, your manganese and your sulfur actually become toxic, and we'll actually see algae growth growing on the soil surface. So those are other things that we can find if you're actually having watering issues. Micronutrients, really quick. The key is to have them in the correct concentration gradient. So a lot of times people in this situation, you might be adequate for iron, but you'll have more iron posted on the report. That is because if you have more manganese than iron available in the soil, the plants get confused actually to uptake too much iron, and will become critical in manganese, and you'll have yellowing because you need manganese for photosynthesis. So a lot of times here, you might find a report, add iron when you don't need it, and it's just to get everything in balance. Also, you need to make sure the iron and copper are balanced. If they're not, they will block the uptake of each other, and you'll have reduced flowering. At the same time, you want to make sure that your micronutrients aren't toxic. Over application of manure for the long term will cause this. Your manganese will be above 50, zinc above 30, copper above 10. That can actually kill plants. So we gotta make sure that there's nothing above those toxic limits. Nitrogen cycle. Everyone's familiar with the nitrogen cycle. Just make sure you have good adequate numbers, right? You want it below, no more than 80. It's wasting your money. Try to keep it around the 20 to 30, maybe 50, depending on your crop. Remember, too much nitrogen will burn the plant. It'll keep it in maturity stage, and you won't get those flowers that you need. Too little, the plant's going to go into maturity too quickly before it has enough growth on it, and it's not going to be able to hold the produce that you're looking for. And you want to spoon feed this nitrogen through, not dump it all at once, else you're going to waste your money through irrigation and leaching of nitrogen. It's the same thing with phosphorus, right? Not enough phosphorus, your plant's going to be stunted, thinning of the canopy, things of that nature. You need phosphorus to grow every single cell in the plant. Without the phosphorus, you don't get the flowers, you can't make DNA, you can't make a cell. Too much phosphorus is actually a problem too. Believe it or not, that is a problem. Too much phosphorus actually blocks the uptake of iron, and that will cause your plants to yellow. And then you'll have to foliar feed iron. And then one quick thing on the soil report, there is a difference between elemental sulfur and sulfate. So a lot of times on a soil report, you'll say, well, why do I add sulfur? I got more than plenty. Basically, the sulfur on the soil report, it's already done its chemical reaction. You're using that for enzyme formation. Elemental sulfur or sulfuric acid is used to drop the soil pH. So it's used for 2 different things. So putting this all together, I'm almost done, I promise. You have a soil report, and laboratory can do the recommendations. I went through so much information, your head's probably spinning, but your agronomist, a laboratory, they should be able to tell you a recipe. This is per 1,000 square feet. It can be done per acre. What elements you need to have for your crops so you know what you need for the growing season. If you're using compost, you can get compost report. It'll tell you how many pounds you're getting in that compost of each element, and then you can put the 2 together and find out how many pounds of compost do you need to meet those nutrient requirements for your crop growth, and that helps you put the whole crop management program together. And I'm almost done, just 2 slides left. They were asked me to talk about the difference between different forms of phosphorus. You have phosphate, which is the plant that the form the plant uptakes. You have total phosphorus, which is the form that people test for in the plant, that the plant uses. And that's usually how recommendations are applied. And then you have P2O5. That is the form that's on your fertilizer label. It's basically total phosphorus padded with oxygen to make the numbers look better. And so if you, for example, if you want to actually know how much units of phosphorus your fertilizer product had, I did a quick math side on the side. If you had a product that was 52% P2O5, you divide it by 2.291, you'll find out that you are actually only getting 22% phosphorus in that product. There's a difference between P2O5 and P. And so if you need, say, 50 units of phosphorus, you would actually have to put down 220 pounds of 0,520. If you recommendations said you needed 50 pounds of P2O5, you actually only need 96 pounds of phosphorus. So just keep in mind when you look at your report, are they telling you need P2O5 or just P? Because that can be a factor of doubling your application rate to get enough phosphorus in the soil for that crop. Same with potassium. Potash is the soluble form of potassium plants uptake. I should say a K, not a P, my apologies. Potassium is what the plant's actually using, and then K2O is what actually is used on the fertilizer. Again, oxygens are padding the answer. So if you actually had a fertilizer product of 0062, in our reality, that is not 62% potassium, it's actually 51% potassium. So you actually have to add more potassium of K2O to fulfill your potassium requirement. So just keep in mind when you look at any soil report, which ones you need so you don't underapply. And again, thank you from our family to yours, IS Laboratories. If you have any questions, please let us know.
Our next speaker is named Dr. Glenn Wright and is a horticultural extension agent for the University of Arizona, stationed out of Yuma, Arizona. He specializes in home and production practices of tree fruits, citrus, date palms, olives, and pomegranates. He also teaches PLS 403, citrus and date production class at University of Arizona. Today, he's going to speak to us about the structure and function of roots.
Hello all, this is Glenn Wright. I'm sorry I can't be with you today, but we're in California harvesting some lemons, and which was something that I hadn't planned on doing on this day when this meeting was going on. So we're going to show you the presentation that I've written about roots, and we're going to go. So we're going to talk about the structure and the function of plant roots, and my reason for doing this is simply that I felt it was important that considering that this whole meeting's about soil and soil health, well, it's all in relation to roots, and so I thought it would be important to get a little bit of basics on the structure and the function of plant roots. And of course, this is approximately 20-25 minute presentation. It'll be impossible to get everything in here that is important about roots. So this hits only the highest of the highlights. But I think it's important to remember the beginnings of roots and the important things about roots and some of the basics about roots, as then you can take this information and keep it in mind as you're talking about soil health. So let's go. So we're going to talk about the kinds of root systems, the Taproot system, the Fibrous root system, Adventitious roots. Then we're going to talk about the functions and structures of the root tip, which have two parts, the root cap and the subapical region. Then we're going to talk about the root-soil interface and how some of these nutrient elements get to the roots through the soil, and then how they enter the root and how they get into the xylem, which is in the root. And then we're going to talk about some factors that control the growth and distribution of roots. And then we'll talk a little bit about growth of roots and shoots and a couple of other things regarding roots. So we have two different types of root systems. Well, three if you count adventitious roots, but we'll start out talking about taproot systems. And dicots have taproots. Dicots are things that don't have parallel veins. They have net-like veins. Many plants, more plants are dicots than are monocots, and many of the vegetables that you all are used to growing, I assume that I'm speaking with vegetable growers, they have taproots. And so this actually is a picture of dandelion, which is a brassica. So brassicas have taproots, dandelion, broccoli, cauliflower, cabbage, things like that. Lettuces and spinaches have taproots. Celery, carrots, beets, tomatoes, peppers, beans, peas, alfalfa, cotton, citrus, which is what I'm interested in, citrus, and olives. And there are many other plants that have taproots that you all are familiar with. And taproot systems develop from the seed, and the origin is the radicle. The radicle is the name for the little root that comes from the seed. And the roots of a taproot vary in thickness. There's the primary roots, which are more thick. The secondary roots, which are, we guess you would say, of medium thickness. And the tertiary roots, which are the least thick or the thinnest. And you basically have one taproot with subsequent branching. And this is based on the assumption that you are not growing your plant originally in a pot. So I work with citrus, and most citrus nowadays is grown in pots, and it will, the taproot will reach the bottom of the pot, and then it will get air pruning. And so you won't have as fully developed taproot in a citrus that's grown in a pot, and I would guess that you won't have as fully developed a taproot with a broccoli, for example, that's grown in a cell that you would if you were to plant it directly in the ground. But nonetheless, in these cases, there still is some degree of taprootness with subsequent branching. And taproots have more vertical growth, I guess you would say downward vertical growth into the soil. They can typically absorb water from deeper areas of the soil than can other root systems. Taproots are typically longer lived than other root systems. They don't necessarily hold the soil particles together, and they can't necessarily prevent erosion, generally speaking, although if you plant them close enough, sometimes, for example, in the case of alfalfa, you can prevent some erosion. But if you're really going to want to prevent erosion, a plant with a taproot system is not necessarily what you want to use. And of course, a taproot can act as a storage organ, such as a carrot or a beet or a radish or something like that. And so here we have a picture, for example, of a 6-week-old spinach, the taproot system of a 6-week-old spinach. And you can see you've got the taproot, which goes down. And obviously, this particular spinach is planted by seed directly in the ground. And then we have a 5-week-old radish. And you can see the radish at the top and the roots that extend from the bottom of the radish downwards. And obviously there's a taproot system, and then that would be the primary root, and then you have the secondary roots that come off from the primary one and the tertiary ones from the secondary ones. So that's the taproot system. And then we have the fibrous root system, and monocots have fibrous roots. So monocots are plants, of course, with parallel veination, and here we have a picture of some green onions. And so most monocots have fibrous roots, not a taproot system. And those would include things such as onions and leeks and asparagus. And many of our grain crops such as wheat and maize and sorghum and Sudan grass and rice and date palms and bananas and things such as that. And I'm pretty much including plants which are grown in the Yuma area, but if you're attending this particular seminar from outside of the southwestern Arizona area, if you're from the Midwest or someplace like that, you can probably think of some other monocots or dicots, for that matter, that are more common in your area. So monocots have fiber root systems, and they vary. They're different from taproots, and they develop from the base of the stem If you look at an onion, for example, the onion bulb is a stem. It's part of the stem. It's not part of the root. And the thickness of those roots is pretty much the same. I mean, there's a little bit different, but if you look at those onion roots, there's not a big difference in the thickness. They don't generally branch too much. They generally clump. They all come out from one area, and they're generally short, and they grow horizontally in the soil. And so, for example, the date growers that I know, we end up transplanting dates, and dates, since they have a fairly shallow root system, are quite successful in being transplanted, because when you go in there with a tree spade, you can get most of the roots from a date palm within that tree spade, because they tend to clump and you get most of them. Whereas if a date had a taproot system, you would lose much of it. So the roots of a fibrous root system for a dicot are generally short. They generally grow horizontally in the soil. There's less vertical growth. They absorb water from near the soil surface, whereas a taproot will absorb water from further down. The roots of a dicot generally have a shorter lifespan. They tend to hold the soil particles together because they prevent soil erosion, because most of the roots are in the upper areas of the soil. There's no storage. The onion bulb, as I mentioned, leaks, and things such as that is a modified stem. You can think of some other plants such as tulips, gladiolus, those kinds of things, which are also dicot or monocot, sorry, with fibrous root systems, and then the bulb itself is a modified stem rather than a modified root. And in regards to fibrous root system, there can be hundreds of them all coming out, particularly with a date palm. There's hundreds and hundreds of roots all coming from the one area right at the bottom of the date palm. And so here we have, for example, a 6-year-old asparagus. And you can see that much more of the roots are more horizontal as compared with the previous photographs that we saw of the drawings that we saw of the taproot system. And on the right, we have a 3.5-month-old onion. And you can see again that the stem, the bulbous stem is the onion that we eat. And you can see the roots below. And yes, there is some downward movement, and there is a little bit of branching, but generally speaking, there's not as much branching, and we certainly don't have a taproot system. This is a classic fibrous root system, so the roots are fairly shallow. And then we have adventitious roots, and adventitious roots are roots that develop from any non-root tissue. So let's back up. So a taproot system and a fibrous root system develop from the radicle, which is from the seed, from the root, the original root that comes out of the seed. Whereas an adventitious roots are plant roots that form from any non-root tissue and are produced both during normal development, such as crown roots on cereal. This is a picture of maize, for example, or nodal roots, such as on strawberries, and in response to stress conditions, such as flooding, nutrient deprivation, or wounding. So these are roots that come from non-root tissue. Date palms have a lot of adventitious roots because we plant dates as basically stems in the ground, and they form roots directly from the stem tissue, rather than planting a date from a seed, in which case you would get roots from the radicle of the seed. But because we plant dates typically as offshoots, the date roots, while fibrous, are typically adventitious roots rather than taproots. So cereal crops, such as most monocots, rely on root systems that are composed almost exclusively of adventitious roots. And I'm going to move my picture down over here out of the way a little bit, and then we'll move it back. And so you can see there are some roots that are on this graphic to the right. You can see an embryonic seed of a eudicot, which has some adventitious roots. And that's a post-embryonic monocot. You can see the brace roots, and you can see some of the lateral roots that come from stems. So, and then the C, you see a post-embryonic eudicot, which in this case is a strawberry, and you see those nodes. and those end up being adventitious roots. And these are normal. And you can think of some other plants, for example, that have adventitious roots, such as Bermuda grass and St. Augustine grass, that spread, and as they spread, they put out roots. And then we have post-embryonic eudicots, which is stem roots, and also some prop roots. And these are roots that hold up trees in more tropical areas. And then there's adventitious roots that come as a response to stress. If a plant is etiolated, which means that it doesn't receive enough light and it becomes long and spindly, sometimes they will make hypocotyl roots, and you can see photographed there. There are some flood or some burial-introduced stem roots, and so, for example, if you happen to take some plants and you bury them, they will make roots, and we can propagate plants in that way. Or there are some nutrient or heavy metal-induced crown or brace roots, which come as a result of stress. And then there's the wounding, which leads to roots. Wounds, herbivory from animals, cutting-induced stem roots. And these are roots that come from cuttings. And, you know, we take a lot of cuttings. Oh, there are many plants that we propagate via cuttings. And how do we get roots? Well, we get roots because of the wounding that occurs. And that actually happens in date palms too. As we take the offshoot off the mother plant, there's a wounding which occurs, and you get adventitious roots from the wound. But we wouldn't be able to propagate via cuttings if it were not for the wound response, which leads to the roots at the bottom of the cutting. So, like I said, cereal crops like monocots rely on root systems composed almost exclusively of adventitious roots. Adventitious roots allow for plant spread, such as strawberries, for example, or Bermuda grass. Adventitious roots allow for the use of cuttings, as I've mentioned, and plants under stress can sometimes survive better because of the formation of adventitious roots. So we'll go on. So we have primary root functions, and those root functions are anchorage of the plant naturally to hold the plant in the ground, absorption of water and dissolved minerals naturally, conduction of water and nutrients to the stem. How do water and nutrients get to the stem? Well, they come through the roots, and storage of reserve foods. So, you know, the plants that make, like carrots, for example, you're storing reserve foods in that carrot, and that's part of the root system. But there's also secondary root functions, and those are the prevention of soil erosion, as we've mentioned before. There is extra mechanical support. So maize, for example, will grow extra roots to support it, or corn, maize will do so. We have nitrogen fixation. Maybe one of the upcoming speakers will speak about that. So legumes have a special organelles in roots which allow them to fix nitrogen or bring nitrogen from the air and turn it into nitrogen the plant can use in symbiosis. I believe it's symbiosis with bacteria. And so that's a root function for those particular plants. There's aeration. Now, many of us don't live where there are mangroves, but if you happen to live in southern Louisiana or southern Mississippi or a place like that where they have mangroves, there's roots that come out of the water, and those roots will provide air. The air will be able to enter the plant through those roots, and those roots come out of the water. We call them knees. Reproduction, adventitious buds on roots. So sometimes you'll put a root cutting in the ground and you'll get buds. So for example, many years ago, we did some work on blackberries, and you can put a blackberry root cutting in the ground and it will grow an adventitious bud. So you're reproducing the blackberry plant by use of by formation of buds on adventitious roots. So this is a secondary root function. Mycorrhiza support, one of the other speakers in this particular talk will talk about mycorrhiza. And of course, regular roots support the growth of mycorrhiza, so that's a secondary function. There is also growth regulator synthesis, so roots certainly do make cytokinins, which affect the growth of the rest of the plant, and that's another root function. And I guess I have to move my picture again here. We have some roots that climb, help plants climb. They help plants float. There are certain plants that are frankly not agricultural plants where the roots have a little air ballasts, which allow the roots allow the plant to float because the roots are filled with air. There are certain roots that lead to balancing, allow the plant to balance better. There are other roots that do some photosynthesis, not many. And there's some roots that do some hygroscopy, which means that they bring water out of the air. And if you're familiar, for example, with Spanish moss or ball moss, how do those plants how do they get water? Well, the roots will help bring water in out of the air. So those, of course, the climbing, floating, balancing, photosynthesis, and hygroscopy are less common functions of roots than the ones that we mentioned previously. So we need to talk about the root tip, and the root tip is about the last 1 centimeter of the root, and we have the root cap and the subapical region. And the root cap is down here at the bottom, and I have to move my photograph again, sorry. It's down at the bottom. And then we have the subapical region, which is a little bit further on, and you can see in the graphic where the subapical region is. And so the subapical region includes the region of cell division, which is the meristem. That is kind of right here. I believe you can see where my arrow is, and it's right there. And then we have the region of cell elongation, which is a little bit further back from the region of cell division. And then we have a region of cell differentiation and maturation, and the formation of root hairs and xylem and phloem, and that's further back. And then we have the primary mature tissue region, which is a little bit further up, not part of this particular diagram, which is where part of it is, where you begin to get the vascular cylinder, the cortex, and the epidermis. And so you can see those items right here. You can see the xylem and the phloem and the cortex and the epidermis. And then you see some secondary mature tissue further up, also outside of this graphic, where you have some secondary xylem and some phloem and some periderm. So the root cap is designated with the numbers 2, 3, and 4 in this particular diagram, and 2 is the columnella. So the columnella is a couple of columns of cells which contain specialized organelles called statocysts. And the statocysts are the cells that help the root sense gravity. They have developed in such a way that the cytoplasm, that is the non-nuclear part of the cell, is at the top, the nucleus is at the bottom, and they produce auxins at the bottom of the cells, and the auxins help force the cells to go downward into the soil, and they sense gravity. Number 3 is some lateral cells that replace the dead ones, and number 4 is the dead cells having been sloughed off. So the purpose of the root cap then is to protect the meristem, and so it's this layer of cells here. It's involved in gravitropism, and it secretes mucilage. So gravitropism is the ability of the root to grow downward to sense gravity. So that's the part of the cell of the root that helps the part of the root that helps it sense gravity. And mucilage is kind of a slimy substance or a lubricative substance which helps the root move through the soil. And so that's part of this. The cells that are down here at the bottom secrete mucilage. Then we have the subapical region, which is numbers 1, number 5, and number 6. Number 6 you really can't see. Number 1 is the meristem. That's where the cells divide, and that's what's being protected by the root cap. And then we have 5, where you have the zone of elongation, which is where if you're doing watering, when you're watering your plant, you're helping those cells elongate. So when you're watering the plant and the plant cells get watered, and inside of them they blow up, similar to balloons. They get bigger, and the cell walls are more elastic. And so when you irrigate and you provide some tension inside those cells will grow. And so when you're irrigating, that's affecting cells in the zone of elongation. That's number 5. And number 6 is further up, you get the zone of differentiation. And the borders of these things are not very clearly delimited. So some of the differentiation occurs closer to the zone of elongation. There's some of the zone of elongation which kind of slips into the meristem area. So it's not like there's a distinct area necessarily where one zone stops and the other begins, but it's still fairly distinguishable. So root hairs. So root hairs are found in the subapical region of the root tip, so not at the very tip where the cap is. This is a picture of a radish seed. Sorry. And you can see the root hairs on there. And the root hairs are a single cell in width. They're oftentimes an individual cell that's quite long and extended. And this is where most of the water and the nutrients are taken up in the absence of mycorrhizae. Now, if you have mycorrhizae, which one of your other speakers will talk about, you'll have more water and what all taken up through the mycorrhizae. But where there's no mycorrhizae, most of the water and the nutrients are taken up through the root tip. Through the root hairs. And this, of course, increases the surface area and allows for a lot more uptake. And you can see how much more surface area there is on this particular radish seed versus if it had no root hairs whatsoever. And the lateral extensions of a single cell, that's what they are. They're a single cell, and they're really only rarely branched. And they're between 15 and 17 micrometers in diameter. What's a micrometer? Well, there's a million micrometers in a meter. So they're between 15 1/millionth and 17 1/millionth of a meter in diameter, and 81 millionth to 1500 millionth of a meter in length. So they're very, very narrow and very short. All right. Now we'll move to the next slide. So how do nutrients move from the soil to the root? Well, they move via about 3 different ways. They move via root interception, which is not only the interception with the root, but interception with the mycorrhizae. They move via mass flow, and they move via diffusion. And then, once the nutrients get to the root, how do they move from the outside of the root to the inside of the root? Well, they move via the apoplast, or via the symplast, and via active transport. So we'll talk about what that means. So, ion movement, ion uptake, how does that happen? Well, we've said there's different ways. There's root interception. And so in this case, the root hair grows until it can reach ions, and exchange then takes place. So here, for example, we have a root hair cell. This is kind of an idealized photograph. This is this white thing that comes out, and you can see that as it grows out into this little crevice in the soil, it will intercept some of the nutrients that it needs to intercept. And, some of those come from the clay particles, and some of them come from organic matter. We're not going to talk about that too much, but clay particles have cation exchange sites, and organic matter have cation exchange sites as well. And those will hold cations or positively charged ions. But eventually, for one reason or other, they'll be released, and they will be available. And so sometimes, not very often, but sometimes the roots can actually just intercept the nutrient that it needs. A second way is mass flow. So soluble ions move to the root with the soil water, and it increases as the transpiration increases. So the transpiration theory, which I believe we'll talk about in a minute, basically means that the water moves from an area of less, how shall we say it, less tension to an area of more tension. And there's less tension in the soil, and there's more tension in the atmosphere. So the water moves from an area from the soil to the atmosphere through the plant. And so that's mass flow. So as the ions are solubilized, they move to the root with the soil water, which happens as a result of the transpiration process. That's mass flow. And then finally, we have diffusion. And diffusion is for elements which are not very soluble. Mass flow is for ions which are soluble, easily mixed with water. Diffusion is for ions which are not so soluble, and they move by random thermal motion, very slow. Fertilizer granules sometimes provide nutrients via diffusion, such as phosphates, which is one reason why you need to get phosphates very close to the roots when you're applying phosphates to the soil. So we'll hit those a little bit more. And this is pretty interesting because this is out of one of the sources that you'll see at the end. And it tells you that for the various nutrients, what the relative importance is of the various types of movements to the roots. So for nitrogen, 98% is due to mass flow, and 2% or so is due to root interception. Phosphorus, on the other hand, there's not a lot of it arrives at the root as a result of mass flow. Instead, the majority is diffusion. And you can see some of these others. And interestingly enough, not all sources agree on this. I was interested to note, for example, that copper, according to this particular source, is primarily brought to the root by root interception. But you'll see that not every source actually agrees with how that works. So then we move on, and you can go back and look at this at your leisure, but just to bring a couple things. So phosphorus and potassium are primary diffusion. So is manganese. Copper is primarily root interception, and the rest of them are either a pretty even mix, or mass flow is the main way that the roots receive the nutrients. Molybdenum, boron, manganese, calcium, sulfur, and nitrogen are primarily moved to the roots by mass flow. So here we have another source, and these sources are listed in the end. And for example, the numbers for nitrogen are about the same, 99% mass flow, 1% diffusion. Whereas if you look at copper, for example, the other one said that copper was primarily made available to the roots by root interception, whereas this particular source says it's primarily by mass flow. And since I'm not a soil scientist, I can't tell you why there's a difference there. But varying sources will tell you the different ways that these nutrients will finally make it to the root. So root interception is the process by which the roots grow through the soil profile and come into contact with the nutrients. And you can see, for example, here's the idealized root tip, and it comes into contact with some of these cations, zinc, manganese, phosphates. That's an anion, actually. And the fact that when mycorrhizae are part of the whole system, then mycorrhizae increases the ability of the plant to come in contact or intercept some of these nutrients. And so mycorrhizae, for example, are bringing in phosphorus many times, and that's because the mycorrhizae bodies, the fungal bodies that are involved there, come in contact with more phosphorus. And so the roots grow through the soil profile, and it is dependent upon the roots actually doing the work and growing through the soil. So if the roots aren't growing, you're not getting as much root interception. So they must physically contact nutrients in the soil. And generally speaking, only about 1% of the soil volume is contacted via the roots, but the addition of mycorrhizal infection to the roots will increase the root soil contact. Then there's mass flow. See, it says mass flow there. And this is the movement of the nutrients in the soil solution to the surface of the roots through the flow of water. And we mentioned this before, but it's a result of the wetting and drying of the transpirational water uptake. It's a result of the stomatal conductance, whether the stomates are open. It's a result of the weather. If the stomates aren't open, then there's no water evaporation from the leaf surfaces, and therefore there's no movement of water through the plant. Therefore, there's no water absorbed by the roots. Therefore, there's no movement of nutrients to the plant via this mass flow system if the stomata are closed. On the other hand, if the stomata are open, then the water evaporates from the leaf surface. The water can travel through the plant. The water is absorbed by the roots, and the water that's absorbed by the roots carries the nutrients to the roots. And so the rate at which the nutrients are taken up by mass flow is determined by the water in the soil, by the amount of water taken up by the crop. So if your soil is too dry, you're not irrigating enough, then you end up not having much movement of nutrients to your plant through the roots. Also, was not mentioned on this slide, but salinity will reduce the amount of water that's absorbed by the roots. Therefore, the transpiration stream is reduced. So if you have a saline soil, you may be getting a lot of salt in your plants, but you're not getting as much of some of the other nutrients that you need. Chelates, when you apply chelates to the soil, they typically will allow nutrients which wouldn't normally move through the mass flow system. Chelates will allow those nutrients to move via mass flow. So an iron chelate, for example, will keep an iron in the soil water solution rather than allowing it to bind to organic matter or a clay particle and be held there. So what chelates do is they allow for those nutrients which are being chelated to be taken up better because they remain in the mass flow system. And then we have diffusion. And diffusion is the movement of nutrients in the soil solution along a concentration gradient. And as crops take up nutrients, the area of lower nutrient concentration is created around the root zone, and then nutrients move from zones of higher concentration outside the root zone into the root zone along the concentration gradient. And so you can see, for example, in the case of potassium, you have an area of high concentration and an area of low concentration. So the potassium moves from the high area to the low area, which happens to be in the roots, and the roots can take up potassium in that way. So that's diffusion. All right, now, once the root nutrients reach the root, how do they get in it? Well, it goes firstly through the root hairs, as we mentioned before, and there's two main ways for water and nutrients, because the water is carrying the nutrients, to move into the root. And one is through the apoplast, and the other is through the symplast, at least for a while. And so the difference is primarily, in a rather simplistic way, that the apoplast is the non-living parts of the cell, and the symplast is the living parts of the cell. So, if the water and the nutrients move into the roots and through the roots through the cell walls, which aren't living, then it's moving via the apoplastic route. On the other hand, if the water and the nutrients move through the cytoplasm of the cell or the living part of the cell, then the plant is taking up water and nutrients via the symplast. But ultimately, all of this water, whether it comes via the apoplast or the symplast, comes to what's called the Casparian strip. And the Casparian strip forces all the water to enter the symplast. So if the water is already in the symplast, then it remains in the symplast. But if the water is in the apoplast, once it reaches the Casparian strip, it's forced into the symplast before it can be taken up into the xylem vessels and thus moved up. So you can see that the Casparian strip is kind of this rust-colored area in this particular graphic right here, and this forces the water to move into the cytoplasm or the symplast of the cells. And then it can then ultimately get into the vessels, the xylem vessels. All right, so here's the symplast versus the apoplast. The symplast exists in the cytoplasm, or the protoplasm of the cell, if you prefer to call it that. That's the living part of the cell. And the water enters the root cells by crossing the plasma membrane. Okay, so it has to move in via probably some transport system. We'll talk about that in a moment. And the symplast is not blocked by the Casparian strip, and the symplast path continues directly to the xylem. On the other hand, the apoplast, which is the not living part of the cell, is the cell walls, and the water and the nutrients move through the cell walls. It doesn't enter the root cells themselves, but it stays in the cell walls. But ultimately, as I'd mentioned, once it gets to the Casparian strip, okay, it's blocked, and it must move into the plasma, through the plasma membrane, okay? So eventually, it all ends up in the same location, that is, to the xylem via the plasma, over the plasma membrane. Well, water and nutrients, I think I'm missing a little bit of this particular title here, but movement of water and nutrients into the symplast or the xylem can be an active or a passive way. So once the water moves into the symplast, whether it's in the beginning or once it reaches the Casparian strip, it must cross a membrane. And this is a representation of a membrane. It's these gray things with these tails. Okay, so this is the membranes are made of lipids or fats, basically, and fats themselves are repellent to water on the outside and attractive to water on the inside. And so these particular elements, these nutrients, for example, have to get across this membrane in order to get into the xylem and being taken up by the stem. So uptake of nutrients is going from the outside to the inside of the roots, and it's made possible by either proteins or other molecules that exist in the root cells, or it can occur passively. So we have passive transport, which is the area here on the left, these blue dots, which are supposedly nutrients, move through across the membrane by passive diffusion. Okay, there's nothing active going on there. There's no energy being spent. Okay, so they go directly across the membrane without any assistance. Then there's another way, which is the one here in the middle. We have another element which is symbolized by these triangles. And these triangle elements move into via what's called facilitated diffusion. That is, there's another cellular organelle, okay, which helps this nutrient move in, but there's still no active use of energy, so it's still passive, okay? So this is considered to be passive diffusion across the membrane. And then finally, there is this active way of moving across the membrane. These are these little squares. And in this case, it takes ATP. Well, ATP is adenosine triphosphate, which is a molecule which provides energy to the cell. So it must use energy in order to get nutrients in there. And so this is called active transport because it actively involves energy. So the nutrients can move across depending upon the type they are and the charge they are and all those things, either passively through normal diffusion, passively via facilitated diffusion, which involves this other cellular organelle, this transport system, but it requires no energy, or active transport, which is another cellular organelle, okay, which exists in this lipid membrane, but this requires energy in order for it to work properly. All right, well, what are some of the factors that affect plant root growth? Well, one is soil composition and texture. We kind of know that, but plants which are grown in sandy or gravelly soils, for example, have a little bit different type of root growth than plants which are grown in more clay type soils. So composition, texture. Flooding. Flooding certainly affects plant root growth. Plants' roots must have oxygen, and if there's no oxygen, then they typically don't grow very well. If you overwater a plant in your yard over and over and over again, it'll eventually die because the roots don't get any oxygen. And the flooding is reducing oxygen to the roots. There's salinity. That absolutely affects plant root growth. Saline water in the soil is more difficult to take up, and roots are damaged via salinity. So typically roots in plants that are saline stressed are smaller roots. There is drought. If the tree or the plant is drought stressed, then there's not as much ability for the cells to elongate, and so roots are smaller in drought stressed trees, which seems to be not surprising and rather counterproductive. It would be helpful, of course, if drought-stressed trees had bigger root systems so they could or drought-stressed plants would have bigger root systems so they could take up more water. That's not the case. Plant root growth. Well, there's mycorrhizae. Well, mycorrhizae aren't technically plant roots, but they do affect plant root growth if you consider the roots and the mycorrhizae as a whole. So the more mycorrhizae you have, the more nutrients you have, and the bigger the roots can be. And we have root metabolism. So aeration is important. You must have oxygen in order for the metabolism of a root to continue. All the things that are going on, the chemical processes in a root must have air. And finally, there's temperature. So roots metabolism occurs as a result of temperature. So soil temperatures can affect root development and nutrient uptake. And I think this is a classic picture from potatoes. So when you have 10 degrees soil, the roots are small. When you have 15 degrees soil, the roots are bigger. 20 degrees soil, and these, of course, are Fahrenheit. So 20 degrees is 68 degrees Fahrenheit. So just to give you an idea, 15 degrees is 59 degrees Fahrenheit, and 10 degrees is 50 degrees Fahrenheit. So 50 degree Fahrenheit, 10 degree Celsius, a soil temperature will make a small root. 15 Celsius, 59 Fahrenheit will make a bigger root. 20 degrees Celsius, 68 degrees Fahrenheit will make the biggest root. But then when the soil begins to get hot, 25 or 77 degrees Fahrenheit, in the case of a potato, the roots begin to get small. 30 degrees or 86 degrees Fahrenheit, the roots are fairly small again. And 35 degrees Celsius or 95 degrees Fahrenheit, then the roots are just as small as they are when they're 10 degrees, or 50 degrees Fahrenheit. So soil temperature absolutely makes a difference on root development. Sometimes I think when we're planting vegetables into the soil in the wintertime, we think that maybe it only affects the upper part of the tree or the upper part of the plant, but the reality is that it affects the lower part as well. Plants can coordinate root growth, and this is a pretty interesting thing that I put in at the end of the presentation. I think this is pretty interesting, very recent. I believe this is from either 2018, 2019, or 2020. But basically what they found out is there is a powerful oxidant which is found at the growing tips of young roots. And this oxidant reacts with materials on the surface of the mature root to produce quinones, not hydroxyquinone, not the drug which may or may not work for COVID-19, but regular quinones. And so what happens is these compounds produce more oxidant production, which creates a signal like a snapshot, which is right in front of the root which is growing, the young root, alerting it to the mature root, okay? So the young root is putting out these particular these quinones, which sense the presence of old roots and therefore it tends to suppress the elongation of the young roots. So the presence of an old root will suppress the elongation of a young root, which I think is pretty interesting. Which is why, for example, roots don't typically get tangled up on each other. Perhaps there is this signaling mechanism which allows roots to avoid each other, which I think is pretty interesting. So there's some sources I want to mention. This is certainly not all of them. I do want to mention the fact that this particular PowerPoint template is found on poweredtemplate.com. And there are some other sources which you might be interested in knowing, and including the one at the bottom, which is the redox-mediated quorum sensing in plants, which is the showing that the roots will avoid each other. There's much, much more to learn about roots. You could spend hours and hours, days and days, month and month, or a lifetime learning about roots, but I've only got 20 or 25 minutes. So thank you for your attention. And sorry, mouth is getting dry. If you have any questions, contact me at gwright@ag.arizona.edu. Thanks.